So today we're going to have a look at introduction and I think it's materials and methods section. And we're also going to have a look at the article, and we'll talk about hypotheses, how to construct them or how to express them in the text. And so yesterday we didn't have time to look at the article, so I'm going to partager l'écran. This one. Ah, ça doit être, voilà. Okay, so can you guys see the article? Yep, it's okay. You can see the article. Cool. So, right, this was the short article that I sent you guys to have a look at. Now, as I said yesterday, it's rare that we have articles this short, but this comes from a clinical microbiology journal, and often medical journals are extremely short because they usually present some results of a test or something. And so I asked you guys to identify the different sections. Now, one reason I chose this article, other than the fact it was really short, is that the different sections weren't identified. So that meant that you guys had to look for them. And most of you found the different sections. So here we have the first part, which is the abstract. Now, this abstract is particularly short. In fact, I would call it a mini abstract because it is just 2 sentences. But, you know, we know what they did, and it shows you how you can be precise and clear in a short and small number of words. I haven't counted how many words are in there, but it must be about 50 or something, maybe less. So that's your abstract. We know what he did. Yesterday we looked at the title. So here we have detection of Burkholderia pseudomyeliae in soil within the Lao People's Democratic Republic. We know what they did. They detected a bacteria. We know where they did it, in soil in Lao. And you know, so it's clear. This is a nice title because it's really clear. There's no it's not too long. It's not convoluted, you know, it's just one straight, what I say in English, dry, you know, there's no adjectives in there. It's pretty much the same thing for the abstract. There's no, hardly any, in fact, there's no adjectives because we have clinical cases of myeloidosis caused by the saprophyte. Saprophyte, it's a bacteria that lives in the environment. Of Burkholderia pseudomyeliae were first noted in the Lao People's Democratic Republic in 1999. In this study, 36% of the soil samples in northern Lao were positive for B pseudomyeliae, providing further evidence for the presence of myeloidosis in this country. It's clear. Now, I have underlined in yellow the different sections as I see them, because it structures out almost as paragraphs. Now, there's one phrase which could be either or, and it's this first sentence here. But let's have a look at this section. So we have a description of what is myeloidosis, a general description. We have some information about the geographical area, some more information about Thailand. You know, you get a lot of cases of this disease in Thailand. And you have some points about who is exposed to this illness. And then we finish with a sentence which tells us that it was first reported in 1999. Then we have a phrase that says, in this study, we have defined, I would have put just we defined, the environmental distribution of bee pseudomallei in soil in the region around the around the capital city Vientiane. Now this sentence here, I would have left at the end of the introduction, but here it's at the beginning of the methods. So, you know, it's a bit of a gray area here, but for me, I would have put it in the introduction. We're going to come back to this different structuring. Then we had the section about the methods. So we had the identification of the sample site. We had the types of methods that were used, where they did it, what they did, how they mixed up the soil, how they incubated it. And you can see this method section is the most, has the most text in the articles, because this is a methods article. We have a short paragraph with some results, and then we have the discussion, which is here, and then we had the conclusion section. Now, after class today, you guys can go back and look at what you identified as the different sections and see how that compares to what I put here. We also have a acknowledgement section. This is really important because often the co-authors, for people who have helped you, but maybe you haven't made them a co-author because you don't think that they should be a co-author, you can put them in the acknowledgement section. And I think that's really nice. Well, I think maybe there's somebody who wants to come in. I have to stop this to have a look. No, it's okay. Sorry. Me and Zoom, we are friends, but not very good friends yet. Well, so your acknowledgement section. So this is important. Often, for example, who do we put in the acknowledgement section? Me, sometimes I reread articles for English or to give comments. So people would put me in the acknowledgement section. For rereading and checking an article, I personally don't consider you should be a co-author. I should be a co-author. So often I'm in the acknowledgement section, and it's also here where you need to thank your funding agencies, because that's really important. And we shall have a look at the results section. That's tomorrow, but here is a map of the area. You know, it's not so easy to follow this map. I have some critique of it. Now, yesterday, Benjamin said you need to learn how to criticize. It's in English, we say more critique, to critique an article. They use a French word for that, in fact, because it means it has a more positive turn than criticizing. So try and keep that in mind. And you can see, we'll just already have some comments here. You see these different, there are different legends. We have some kind of circle with a point, we have a filled circle, and we have a circle with a cross. But, you know, this is a PDF that I downloaded from the site, and you can see it's kind of not that good quality. So when we will talk about presenting your results, keep that in mind when I say you need to make sure that the graphic, the image, the graph, the table, whatever, it will reproduce well on the screen. And here, you know, if I didn't know what the eastings and northings, what the latitude and longitude was of this area of Laos, I wouldn't be able to read that. You know, I think it's like 104, 103, but you know, you have to be careful about that. It's not. The problem is when you do figures and graphs that aren't very good, that aren't good quality, you will lose the attention of the reader, and you don't want to do that. You want them to keep reading and to think about what you did and to cite what you did. So anyway, And the other point I wanted to point out what the references. Now, on Friday, we will talk about references. And one thing that is really painful with publishing articles is that each journal has their own format because, you know, everybody has to have their own thing. And I think it's really stupid, as you can tell, but you don't have any choice. You have to do it, because I have had articles that I have submitted and it has been rejected because the references weren't in the right format, because I have used a reference format that was up to date 3 years ago, but they have changed their format since. And there is, somebody wants to win. There we go. Yes, the different reference formats. So before you submit, go to the website of the journal and see what their format is and download it into either your Zotero, your EndNote, or whatever. And because it will save you a lot of time. I'll show you on Friday why you should use these things. That's because if not, you will find yourself right here. You can see, or look at number 6. We have the name of the person, their initials, a point, a comma. And this guy, his initials, they are after his name. But the second author, it's before. But for some journals, the name will be after, and it will be, it won't be in bold, it will be in normal text, and the date maybe it'll be in brackets, and it's just so annoying to change all of that. So keep that in mind. And that was one of the questions I asked. And also the way in which we cite inside the text. This guy, it's numbered inside brackets. For some, it's a number in a superscript, and for some, it's the name. It would be bibliography here. But, yeah, so these are small things, but they're not. That's what the trap is. They seem like really small things. They don't seem important for the article, but article is also a lot about format. And that's one of the ones that can be really annoying. Okay, so let me stop that guy and start the PowerPoint introduction. Da-da-da. Put that there. Let's try this. We're going to try on full screen. Oh, no, no, we're not, because it won't let me. That one. Okay.
It works.
Right. You know, it's not me, the master here, it's Zoom. So you see introduction. You don't see material methods, because I have both open. Okay, so we shall start the introduction section. Now, yesterday, we had some questions. We talked a little bit, I can't remember who it was, that said about the introduction. We had some comments about that. So the introduction, what you need to keep in mind is that it introduces the subject. It sounds really obvious, but it doesn't develop it. And the development is going to be in your discussion, but you need to present it. So you need to know what you're doing and why. And your main objectives of this first section are to capture the reader's attention, because, you know, they need to know what you're doing, why you did it, what the big picture is, why is that interesting. It's not just that I felt like going and measuring some soil in Laos, because Laos got some really nice soils. And you need to give enough information to the reader that they don't, oh yeah, that's really interesting. I'm going to continue reading, and I can see why this is important. So keep these ideas in your head. You need to, one thing is when you're writing, you always need to keep in your head, what do I want the reader to learn? What do I want to transfer to the reader? And is this interesting for the reader? I think maybe we get a little bit trapped in the idea that we need to write because we need to write, and that's it. You know, we have to publish because if not, we don't get our grant, we don't get our salary. That's not the real reason why we publish. The real reason why we should be publishing is to share the information. So, now the introduction, it sets the tone for the paper, it sets the quality for the paper, and it's when the writer, so you guys, begin to determine if the reader will continue reading and if he, whether he's going to find it satisfactory or whether he's going to think, it's not so good this, I'm not going to continue anymore because their introduction is badly structured, I don't know what they're doing. It's yes, Benjamin.
Oh, thank you. You see me. Sorry to interrupt, but I just wanted to testimony and confirm what you're just saying, because I've had the opportunity to evaluate many projects. And when you start reading something, really, the first page is key, because even if it's not explicit, in your mind, you go in one way or another very quickly when you start reading a text. So for the reviewer, the introduction will be really very important. You need to spend a lot of time in the introduction because it will lead the opinion of your reader very quickly. Thank you.
Totally. I wholly agree. And there are three things that when I read an article where I'm not the worst of reviewers, but some things really annoy me. And I'm no different than anybody else. The things that are important is you need to make sure that the title is in correct English and that it is well structured. This is the same for the abstract and for the introduction. And I would go as far as to say the first paragraph of the introduction needs to be perfect. Because if I start reading and at the end of the first paragraph, it's like what I call spaghetti, you know, the sentence is going all directions. It's going to put me in a bad mood. It's going to irritate me and I will be particularly harsh, or more harsh than normal, reading the article. And so I really encourage you to get somebody to what I call clean up your titles, your abstracts, and the first part of the introduction, and if possible, all of it. Okay, yes, totally agree. Where are we? Nearly next page. Yes. And so what's in this introduction that has to be perfect? Well, you give a short review of the main subject of the study. Remember in what we saw on the PDF? You know, Crocodile pseudomalia was found in Laos in 1999. And so, you know, we're wondering what it's like now, 10 years later. The problems, the limitations, and the shortcomings of existing literature. This is where we come back to that question of you need to know the literature in your subject, because if you don't, you can't present the limitations, the problems, and the shortcomings, because maybe in Lao, you know, there were only that first article from 1999, maybe they only measured 3 samples, 3 soil samples for the whole of Laos. It's nothing. So this would be a limitation. Maybe the method they used before wasn't so good. So this is a problem or a shortcoming. So these are things that you need to present as well. You need to give information on the aim of the study. Well, you wanted to see what the geographic distribution of the Berkhold area was in Laos, and the type of study. Well, it was a field study where you went to sample. Maybe it is a study of silting in Haiphong Harbor. Maybe it is a study on access to wastewater treatment in Hanoi, in comparison between Hanoi and Ho Chi Minh, a comparison between Bangkok, Phnom Penh and Ho Chi Minh. I don't know, these things. You will say what type of study it is. So this information has to be there so that at the end of the introduction, we know why it's being done, what already exists, and what type of study, literature study, modeling study, socioeconomical study, etc. You guys know what you do. You need to go to the heart of the subject. It needs to be clear, and it needs to be concise and, if possible, interesting. Avoid boring, avoid blah, 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 pages and pages and pages. Oh, no, nobody reads that anymore. We have so much information now. Articles that are short and targeted and clean and don't have blah, 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 and don't have a lot of adjectives, people will read them. You guys know, because when you get an article, if I had sent you an article of 24 pages, you guys would have read the abstract and the title and maybe the conclusions. That would have been it. So it's the same in normal life. Ah, somebody's coming to join us. Voilà. So keep that in mind and try not to do something too long unless you're doing a systematic review. Systematic reviews, of course, need to be long because they're systematic reviews. You should be up to date in your literature. This is something really important as well. And now I know you guys often can have difficulties accessing up-to-date literature, but you guys will have the letter for writing to corresponding authors. And also now with open access, things are becoming much more democratic in terms of access for articles. And try to keep a historic of the work. Don't just cite things from 2017, 18, 19, 20. Make sure you can have a short timeline of the study. The article from Lowell, 1999, the first sample. So, and in general, the introduction is several paragraphs to several pages. Now, in the article I showed you, it was 1 paragraph. In an article that is a little bit longer, maybe you would have 4 paragraphs. Sometimes a bit longer, but I think 4 paragraphs you can present the background clearly because you can divide it up into the different sections. In general, I'm on the 4 to 6 paragraphs for introductions. This is what I try and do. I don't like writing really long articles. I find it's not easy. It's boring. It's boring for someone to read. It's boring for a reviewer to read. And when you get bored reviewers, in generally you get bad results at the end. They reject it. Okay. Oh. Right. So your objective, you need to convince the reader of the importance of your study. You need to give reasons for investigating this particular subject. If we look at we think about the PDF, what we had was that Burkholderia pseudomallei, it's the pathogen for melioidosis. It's a disease that is endemic in Laos. And so we need to know where people are getting it, because you see it in farmers and agriculture, farmers and fishermen in the water. So keep that in mind. And your study should answer a timely and important question. And this, of course, is something that happens before you start writing, but you need to keep that in mind in the way that you present it. It needs to be clear to the reader that this is something that's important now, or is important, but particularly so now, because it will capture the interest of the reader. Your rationale of why you did it needs to be strong, not just it's important. You need to explain why. Important for public health in the case. And it should be an addition to the existing literature. Remember yesterday I said that it was important to know your literature and to do new things. That's what a publication is. You publish new data, a new opinion, new data, a new review. You need to try and avoid repeating the same thing and saying the same thing. Now, if you are doing a comparison of old data sets or formerly published data sets and you have a new vision for that data, I think maybe that can happen quite often in social sciences, where you have a different way of presenting the data from a data science article. It still has to be new. Don't just do the same thing, because there is a certain chance that it will be rejected. Because you'll say, it's the same as what Bouclet did in same site, same data, same opinion. What's new? And you spend so much time writing, you know, it would be a pity to waste that time and not get what you wanted in the end, which is, of course, the publication. So, how to do it? You set out the scope of the study. It was Lao. You give a quick overview of what's going to follow. When I say follow, it's what's going to come in the next part of the article. Your introduction, as I said, must be short and to the point. Think about the first, the PDF. You know, you don't get much shorter than that. And it should be easy to read. And don't use too many references. Now, it sounds contradictory with what I said when I said that you need to know your literature and reference your literature. But if you look at the PDF, you will see there are maybe 3 or 4 references in the paragraph, maybe less, maybe 2 in the introduction. I don't recommend you do an introduction with only 2 references, unless it's an introduction of 1 paragraph. But don't put in 40 references. I have seen cases where people have, a phrase will be, climate change has led to increased rain precipitation and intensity in the northern uplands of Laos. And there will be 6 references. Try not to do that. As a rule of thumb, I say 3 references, max, per phrase. Some journals limit the number of references you can do. They will limit to maybe, what is it? When you have 5 pages of text, it's about 50 references or something. Limnology and Oceanography, which is a journal that I knew, they have a limit on the number of references. If you have review articles that you can cite those. You can see the review by Bouclet for this. You can do that. Now, that's not a problem. And but keep in mind those references, because you're going to maybe reuse some of them in your discussion, because you need to link the introduction and the discussion together, but we'll come to that Maybe tomorrow, I think. I can't remember. Keep the structure simple. Begin with a very general statement, and then you are going to be narrowing down towards the thesis statement, the hypothesis statement, in the introduction section. You will give a general statement about the subject. Bacillus pseudomyelinae is the pathogenic agent of melioidosis. Beginning to focus. This disease is endemic in Laos and northern Thailand. More specific, in 1999, the bacteria was first detected in the soil. And down again to, we hypothesize that the bacteria is present in sediments and soils because most of the patients have been farmers, rice farmers. So you can see how you're coming down, you're coming down, you're coming down. You're starting very big, sort of a global vision. You're coming down to maybe a regional vision, coming down to a countrywide vision, and we have somebody, excuse me. Voilà. And then coming down to the real question, maybe what you're going to say is it's in a specific type of soil and you want to investigate that. One way of visualizing this is thinking about a funnel. Because the funnels are, I think this is a visual way of looking at maybe the intellectual dimension of each section. And one thing that I recommend is that when you're going to be writing your introduction is to put these subheadings here. Have a section that's called General Introduction, have a section that will be more specific aspects, regional aspects, or however you want to structure that, whatever your next degree step down is. The subject statement, what you were doing, and then the hypothesis and objective statements. Now, I don't say that you should keep these subheadings when you're going to submit, but in the beginning for writing, it will help you place your phrases in the different subsections. With Word, now we can slide phrases around. Before, you had to rewrite it and everything, and it was a bit painful. But with word processing software, it's so much easier. And I really, really, really recommend using subheadings throughout the whole writing process because it helps you structure. Because one thing, I think the thing that I find is perhaps the most difficult when you start to write, it's the structure. I already talked a bit about spaghetti. Spaghetti is when, you start talking about something and then it goes off and talks about something else. And then the first subject comes back and it's really hard to read. And it will get your paper rejected if you don't have structure. What you need to go for is what, somebody else? Attends. Voilà. You need to go for something that is layered, like like the Italian food lasagna, where you have different layers. You have a layer of introduction, you'll have a layer of methods, a layer of results, a layer of discussion, and then inside each layer, you will have the different sections written in order and not going back and doing circles and things. Try and keep that image in your head when you're writing. Okay. So, introduce your subject, brief description, what is the experiment, what phenomena are you studying, and a brief history, and explain how it relates to your current research. Summarize the previous research. So, well-rounded, that is relevant to your topic. Don't go off on, some kind of intellectual discussion on subjects that are so far away. Don't do that. You'll lose your reader, and they'll probably, your reviewer will say it's irrelevant. Before you write, thoroughly research your topic. journal articles, internet databases, you know, like in health area, you get lots of stuff on the World Health Organization website. There's documents, but be careful what you're citing. Make sure you cite things that have been peer reviewed in one way or another. Sometimes, you know, newspaper articles can be cited when you want to put forward a point, but don't use those informal databases. I call them informal. Non-peer reviewed literature sources too much, because you know you need to be able to verify the quality of the article, unless of course your discussion is about the quality of the article. You see what the difference is. Take notes when you're writing these articles. You know, make sure that you keep track of the sources, and to avoid plagiarism, you need to, if you're citing somebody else, you need to cite them correctly in your text. And you need to provide your hypothesis. So yesterday, I asked you, or we asked you, to provide your research question, to identify your research question, and also to structure, to give your hypothesis in a phrase. Because I wanted to see how you guys, well, your subject, and also how you structure your hypothesis. And so, in your funnel, you're coming down, you're in the last, the second to last and the last section now. You have summarized the previous research, and normally, if you have well constructed those first, second, third paragraphs, it's like obvious what you're gonna do, because you have made like you know, it's like when you're herding sheep, you push the sheep towards one gate, so they have no other choice. We have someone, conversation. I can't see.
Well,
Benjamin.
it is a question that I received by email, which is quite interesting, and that is relevant to what you're just saying, and it is related to the hypothesis, formulation of hypothesis in the case of a qualitative research for like ethnography or interviews or things like that, which is aimed at understanding a phenomenon, suggesting that qualitative research has a central question and associated sub-questions, what we call generally a problematization, problematization in French. And the question here is that in this case, should we formulate hypotheses and questions as in a quantitative review paper? I have my opinion about it, but maybe Emma, you are quite used to transdisciplinary researchers, and you have your ideas about it.
I say yes, for me, because having a hypothesis is, you need to compare something against. You need to compare your data against something. And so this is how for me, yes, but I don't know, maybe you have a different opinion, Benjamin.
I think
ethnography, I haven't really read much ethnographic text, yeah. So I can't.
I will give my point of view. I will give my point of view, but I would be very happy to hear from trainees what you think about that. From my perspective, and according to my experience with human sciences, I will be maybe a bit harsh here, but I think it's laziness from a human scientist not formulating hypotheses, because it is a trick. I mean, it is tricky, and it is a trap actually to save this energy, because of course in a qualitative question you have a central research question and then sub questions, which is a way to build up the issue, the problems. But even with sub-questions, you should have hypotheses. Because if you don't have hypotheses, how will you justify doing research and doing activities, implementing activities to check if your hypotheses are, because in any way you do have hypotheses, you do have ideas of what you might think. Otherwise, you can't elaborate any some sort of research protocol or methodology. The definition of your methodology is related to some hypothesis. Because if you have this hypothesis, you will use this kind of methodology. If you have this hypothesis here, you will use another kind of methodology. So you have a hypothesis, and I think it is important to be clear about that. But I suppose my opinion is not shared all around the world, because we read a lot of papers in human sciences which do not expose as hypotheses and are not clear at all about it. But I think it's a mistake. It's also because of that very often papers in human sciences are hard to read, because you don't really understand where the author wants to go, and you don't understand the questions, the problematic, etc.
It you know, it can be the same thing in data science. If you don't have a clear statement of the hypothesis, you will have difficulties structuring the article, because it becomes you know, say in français, balade dans la forêt. You know, it's a nice, having a nice little walk in the forest and going, and we can't do that anymore. And this was done in, you know, like the 1800s. Naturalists would go and look at stuff and think about things. It doesn't work that way of writing anymore, and it leads to things that are just blah 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 blah. And the problem is, in blah 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 blah, the real subject, the real response is lost.
Yeah.
And you know, it's a waste of time, and it's really sad, because I'm sure in the middle of blah 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 there are some really nice information, but if it's not accessible because it's just buried, you don't know. This is for me why it's so important to have a clear hypothesis, because it helps you structure your work.
Yeah,
that's why. And
I
agree with you. It applies to everything. Maybe you don't need to write the phrase "our hypothesis was." It can be "we hypothesized that," or "the central hypothesis of this work was that." You know, you can. It doesn't have to be such a.
Or our insight is that or
yeah.
We suppose that. Yeah,
I agree.
Kong had a question about where we can find hypothesis in the article you sent.
Right, this guy, they don't state it exactly, but I have a slide that's coming up next, or this one, the next one, or the one after, where I show you exactly that, because those guys, they should have written it, and their hypothesis was, for me, their hypothesis was that there were that the geographic extent of Berkholderia pseudomalaei was much larger than what the first article had shown. But it wasn't me, the reviewer, for that one, so you know. So we'll come to the hypothesis. So these are the things you need to keep in your mind when you write your hypothesis. You know, what is missing from previous studies? What research questions have yet to be answered? In fact, it's that one that's really the center of how you can structure your hypothesis. And it comes at the end of your introduction. We give it, and then sometimes you can describe what you expect to find in your experiment or study. This happens quite a lot in environmental work. So, yes, I changed, so let me go. So what is the format of a written hypothesis? Well, you can have bacterial growth may be affected by moisture levels. What I would have put, bacterial growth is affected. Excuse me. Bacterial growth is affected by moisture levels. Where is my mouse? Is there? Bacterial growth is affected by moisture levels in the air. Ah, Ketia is there. Ah, look at that. I found the conversation. Cool. So, right, here we have a statement. It's a hypothesis statement. Bacterial growth is affected by moisture levels in the air. Monocultures of maize increase soil surface runoff. They are statements, they are positions. They are statements that can be tested. Mm-hmm. Ah, we have someone else who comes. In other words, you know, so you have to be testable. You have to be able to look at your data and say, yes, this is true. No, this is not true. You can use statistics for that. We'll come to that section tomorrow, I think. And sometimes the statement will be written as, we hypothesize that teak plantations on steep slopes lead to increased soil erosion. So, you know, we know that these guys, they're testing, is the presence of teak a factor in soil erosion? So you can see, you could have, I don't know, climate change has, increased precipitation in the Red River Basin has led to, is responsible for silting in the Achtang estuary Haiphong. You can test it. Is there more rain? Is there more silting? Yes, and you can look at the different factors that influence, but it's something that can be tested. It's not just, oh, we wanted to see if moisture was impacting, we wanted to see how, what was affecting bacterial growth? Ooh, you know, there are so many factors and it's not very precise, so it's difficult to test. Voilà. So here is an example of what we did, the hypothesis, and this was in the last paragraph of an introduction. So the objective of the study was to assess land degradation relating to the export of organic carbon and soil bacteria, blah, 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 blah. We're coming back to teak. Again, it was some work that was done in Laos, up in the north of Laos, near Luang Prabang. We say, so the first, we have an objective. We say how we did it. Rain simulation, here. And we have the hypothesis, and you will note there are two parts to the hypothesis. We hypothesize that A, teak plantations led to a higher export of SOC, soil organic carbon, and bacterial blah, 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 in runoff. And then that between the two teak plantation practices, one was less erosive than the other. And so this is the end of the introduction. We know that the reader knows that two things were tested. We tested whether or not teak plantations were inducing erosion of biodiversity and soil. And we tested that between the two teak plantations, is one worse than the other? Kind of making a shortcut. But what you guys can do, if you want, you can take that paragraph and you can change the words, but you can use the starter phrases here. The objective of this study was to investigate whatever you were doing. We hypothesized that, whatever you were doing. So you can use that format. And that makes your last paragraph of the introduction, and you're down to the point of the funnel. And we're ready to bounce on to the methods section. Now, because I want these classes to have a certain pragmatic orientation, when you submit your article, it gets reviewed, and it will get reviewed by the editor, maybe 1, 2, 3, 4, or 5 reviewers, depending on how enthusiastic the reviewers are. And the things that you will ask as a reviewer when you're reading an article, the first is, are the objectives clear? So, well, why it's important to have the clear statement, hypothesis statement, objective, hypothesis statement paragraph at the end. We have, is the importance of the study adequately, but also just correctly emphasized. Don't oversell something because they're, well, you know, it's a bit odd that why they're insisting about this, but make sure that it's adequately emphasized and you cite the literature, previous literature, correctly in support. Is the subject matter new? Hopefully, yes. And also, is the previous work on the subject adequately cited? So these are 4 points that I will keep in mind when I am reading an introduction. Also, is it structured correctly? Is it well written? And these are the important parts for me. So we have a couple of slides about the title, and then we'll have a short pause. So your title, it needs to succinctly describe the content of the paper. You need to use words that are strongly associated with your paper. So for example, the organism in the PDF, it was Burkholderia pseudomelia. The location of the field site, it was Lao. You know? And remember yesterday I talked about keywords and the title. Make sure you use strong words, words that are strongly linked to the subject, so that these database researchers will capture your article, because more and more we're using these database electronic database searches to find articles, and so it's important to really think about the title. It's not really a section, but it's necessary and it's extremely important. And as I said a few minutes ago, make sure it's in nice, clean English. It should be short. It shouldn't be long, but it shouldn't be too short that we don't know what's happening. And you need to make sure that, I think, it's really important to have those words in there. I don't necessarily think you need to put in regional, you know, like in your title, although the PDF they put in Lao, that for me could be a keyword maybe in most instances, but that particular article was specifically related to soil in Laos, so it was important. But in many of my articles where I do the work in Lao, it's not in the title, but in the keywords. So some examples of titles that are maybe not that strong. One would be fruit fly behavior. Okay, you know, it could be anything. It's short, it's succinct. There's no comment about that, but it's too short. We don't know what's going on. Another would be maybe the effects of an organic pesticide on the feeding flight behavior in fruit flies. Well, we know what's happening. We know it's a fruit fly. We know we're testing organic pesticide. But maybe you could redo the title in a more active way. Organic pesticides reduce the speed and duration of feeding flights in fruit flies. I like this last one. sometimes people don't like putting such strong statements in the title, but I think it's more attractive. I'm more likely to read something that's got a title, this last title, than something that's a bit, the effects of an organic pesticide on the feeding behavior of fruit flies. It's much longer. I think when you read it, you can see the difference in the rhythm of the title. And so, right, this is the last slide of the introduction section. Now, I see there is a question. How can we identify or state the research question or hypothesis in other articles? Are there any specific principles on how they use? Often the research question will be the objective of this work was. The objectives were to, you have keywords like that that highlight what the objective is. Or we hypothesize that, the hypothesis is. Normally it should be very clearly stated, and so it's relatively easy to identify. If it's not clearly stated, I would say, in my case anyway, that it's not well written, the introduction. And so sometimes you have to read the whole article to like divine. You have to, what is it? I don't know, maybe it's that, to find the hypothesis or the research statement. And that's just a bit painful. And you will find that when you can't identify what they were doing, you will read less and less of those articles. And so, when you guys write, don't waste your time. Don't, make it, state the research question, the hypothesis clearly, and more people will read your articles. The better the article is written, the more it will be read. It's as easy as that. So, I am going to stop there now, and we shall have a short break because we've been going for an hour now. And then when we come back, we shall look at the material and methods section. So we'll go back to the PDF to have a look at that in a bit more detail. And then we'll talk about some slides. Okay?
All right. Thank you very much, Emma. So 5 minutes break for a short coffee.
Yes.
See you in a bit.
See you in a bit.
Here.
Okay.
Okay. So let's start again.
Yes. Right. So we just finished having a whirlwind tour of the introduction. And now we're going to have a whirlwind tour of method sections. And so we shall go back to the PDF. Let me put that on partage d'écran. This one. Voilà. Okay. So now we're going to have a look at this part from here, all the way down to here. And so what we see in this study, we have defined the environmental distribution. So for me, this is an it's a statement of what they did. I would have put that the objective of this work was to define the environmental distribution of B. pseudomeliae in the region around the capital city. And so it would have been in the first paragraph. But as I said, it wasn't me, the reviewer. And then we can start the method section. Now, my yellow underlining has included the A, but that's it's because I did it badly. Normally the sentence starts with the A. A survey of rice fields was undertaken during September, the rainy season, along roads radiating out from Vinciane. So we know what they did, very general terms, but we know where, we know when, September 1998. We know where the sampling sites are in the figure 1 that I already commented about, the I think is a bit poor quality. And they sampled in rice fields that had been recently plowed or planted. So we know. No references in that paragraph, you will note. Then we have a description of the technique, the qualitative analysis that they used. So where they tell us what they did. They had the soil samples, where they came from different locations, what they took, 3 cubic centimeters of soil. Soil is measured in volume in general rather than weight. How they took it, all the different little steps that were done, taken back to the laboratory, how the samples were prepared, how the samples were incubated, and so forth. Now, if you had access to the products that are mentioned and the protocol, the one of Ashdowns-Ager, the guy here, this is the method they used, you would theoretically be able to repeat what they did. And this is important for a method section. And remember, we said yesterday that research should be repeatable. You should be able to take an article of somebody else and do, if you have general knowledge in the study area, and redo what the person did, what the authors did. They also describe in the second paragraph a quantitative analysis, how many sites, where they took the samples, how they did it, how it was transported to the laboratory. Again, the step-by-step treatment of what they did. However, it's not in painful, what I call painful detail. Painful detail would have been, we took the sample and then we shook it 15 times and then we moved it 4 meters. And you need to, when you're writing this part, you need to keep in mind the difference between an article and maybe a thesis chapter. Because sometimes the thesis chapter, you have to go into intricate detail. Well, here you don't. There should be enough that somebody can do it, repeat the experiment, but not so much that they get bored reading it. So this is one example of a very short method section, but it's probably repeatable. I have another one that I want to show you guys. Before we move on to the PDF, I will send this PDF to Benjamin for you guys. Where are we? Partager l'écran. Here we go. Okay. Now, I talked in the first article, we had a text-based description of the methods. In some cases, you can complement that, accompany that text with a graphical image of what you did. Now, if you have several steps, it's a flow chart you could use. If maybe you're looking at cohorts, you're discussing cohorts of people, you can say how they were selected, whether they're male or female, what their different age groups were. Sometimes a flow chart makes that much easier to follow, so you can describe which data you kept at the end. And here is an example of an experiment that we did, where we incubated water in greenhouses and then in experimental lakes. And we describe, so here we have the pictures of the different greenhouse experiments with the different measurement variables, the experimental lake variables, the measurement variables. You can have nice little diagrams like this. When we tried to write this in text-based format, it was really hard to follow. And so one of my co-authors suggested that we do a schematical diagram. And it's true that if you understand, if you're familiar with this type of experiment, it's much clearer. So you can keep that in mind as well. Sometimes a schematic, schema, conceptual diagram even, it's much clearer to transfer the message of your method section. Okay. So I will give that to Benjamin and you guys can get it. And you will be able also to have a look at the structure of the article. It's one of mine, and so you can probably critique it if you want. Not even probably, you can critique it if you want. So, material method section. So, materials and method section, you have several parts. The full description of the materials, study site, for example. Full description of the methods, what they did in our PDF example of Ashdown's media. Description of the type of study, and my favorite friend, the statistical methods, because you know, I'm an oceanographer, and oceanographers don't like statistics, and I'm one of them. But nowadays, we can't get away with it, so we have to use statistics. I shall talk very briefly about that tomorrow, but I encourage you to seek help from somebody who's really good in statistics. So we just looked at the methods in the short article, so we can move on. So you need to explain clearly, the key word here is clearly, how you carried out your study in using the following general structure. Now this is for data papers, but you can also apply these ideas for other types of articles. So you would talk about what organism you studied, if it's relevant, how they were treated. You will say where and when the study was carried out, but you know, this is only if location and time are important factors, because sometimes we will do an experiment in the laboratory with laboratory-based cultures or something. It's not important to say the experiment was carried out in Emma's lab in Paris in February of 2000. That's not important. But in the case of the article that I showed you, it was important to say it was in September, because it's the rainy season, the end of the, well, coming towards the end of the rainy season. If you're working in the port of Haiphong, it's important to say it was conducted in the Haiphong port in I don't know, July, when Red River flow is high, or if you're looking at a study of the impact of the COVID epidemic on schooling of children, you would have to explain the period and what age of the child and the things like that. So, you know, adapt it to the situation, but give the information that is necessary so we understand where and when. If you do a field study, you need to give the description of the site, significant physical and biological features, and the precise location. So to come back to the port of Haiphong, you would give the location of the port of Haiphong, specific physical features, the form of the port, the depth, the dredging frequency, things like that, if these are important factors in what you did. You would explain how your study is structured in terms of the control. What controls did you use? What treatments? What variables were measured? How many samples? How much replication? How the data is presented. Maybe you went and did semi-directive interviews with people, asking them, I don't know, what type of wastewater treatment they have, what do they use, where do they get their drinking water from, how do they how do they perceive the water quality, the wastewater treatment systems, things like this. You will have all of these this data that you will have collected. So this you would present, you know, this study was conducted in a small village in northern Thailand, and 52 people were interviewed, and the data have been collected and split into age groups or male and female. You will describe that. It's important to know. And you would say the final form of the data are presented as means or as aggregated data for each age group or something like that. You need to say what the protocol was for collecting the data. So semi-directive interviews, or dredging, or data were collected from the maritime agencies, or from the urban planning department, things like that. What the sampling techniques were used, how the samples were collected and stored. This is important when you're dealing with biological samples. And if you don't write this, it's one of the first things the reviewer will ask for when they start reviewing the methods section. How were the samples treated? How were the interviews conducted? Was there the ethical statement? Were the populations, did they have full disclosure on what you were going to do with the data? These things like that, they need to be noted. You will say how your samples were prepared if you have analysis, you know, the chemicals and the methods that were used, your units. I can't remember, I don't think any of you guys do analysis for your chemical analysis of your data, but maybe some of you who are working on water quality and wastewater, it's something that's going to come up. And so once you have all of that information, you need to structure it into paragraphs. And before I said use the subheadings, in the methods section, it's a section where we often use subheadings to separate the different sections. And I really, 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 really recommend that, because it helps. In fact, I shall show you the other PDF. It's really interesting when you have longer articles. The PDF that I sent you guys, that we sent, it's too short to have subheadings, and some journals don't like them. But if we look at this one, this is the one with the, where is it? This guy. You know, it's the same article. And you can see material method section, introduction, talk about our figure. We have subsections. We talk about soils. We talk about the experimental lakes, the aquatic microcosms. We talk about the different measurements. We talk about bacterial abundance, metabolism, and, oh, my favorite section, statistical analysis. Now you guys can do this in your articles, and even if the journal doesn't like or doesn't ask for subheadings in your methods, I suggest that you do it, because, you know, it's like a pen, you know, like a pen where you put animals, not a pen that you write. And you can put all references of the methods, all statements that concern the methods into that paragraph, structure it. All that considers method 1, they go into the paragraph, and they're not elsewhere. So it helps that lasagne thing, you know, or tiramisu in the different layers. This way you avoid spaghetti, because spaghetti is bad in scientific writing. Eating spaghetti, of course, is good. So let's come back to my PDF. So as I say, use the subheadings, and each experiment or procedure, you know, you split it up into different sections, even if it's broken up over time. So it's not, you will not necessarily present your methods in the chronological order. They will be in a more logical order. All of the chemical analyses will be together. All of the statistical analysis will be together. The sample site, the sample study area, the village where you did your semi-quantitative, semi-directive interviews, you know, that information will be together. Maybe you went to visit 6 or 7 villages, but over the period of 18 months. And so the first village you treated, you got your data, and then you treated the data. And then you went to the next village and you treated the data, and so on like that. But don't do it like that. Do it in an order that is by action and not by time. If not, it will be difficult to read. And make sure you provide enough quantitative detail so that other scientists can do the same thing. Obviously, with interviews, you know, they're not going to get the same answer, probably. As you know, human perceptions will change over time, but the way that you approach the subject, the way that you studied the subject, that should be clear. Often we can be a little bit too economical in the method sections, and sometimes the reviewers, they will ask for more information or clarification. When you present the methods, if it's a new method, you need to describe it. If it's a method that is already known and very common, use the reference. Don't go into painful detail for methods that have been known for 30 years. You can save the space, because if you're in an article, if you're in a journal, or you want to publish in a journal where you have a limited number of words, well, you know, maybe it's wasted words, and you might need those words elsewhere, like in your discussion. So I recommend, like, I don't know about, maybe you will be using a semi-directive interview technique that is well known in your field, that Benjamin published 15 years ago, and everybody uses that technique. And so you will say the semi-directive technique of described by Bouclet et al. In 2000 was used for these interviews. However, if it's you that has developed this new technique, and it's been developed from the very beginnings by you, you will have to describe how you go about it. If it's the technique of Benjamin that he described in 2000, but you have adapted it for the situation, you will say the semi-directive technique of a semi-directive technique adapted from Bouclet et al. 2000 was used. I don't know, maybe you decided you only wanted to talk to the female heads of the household, and so you had taken a subsample. Well, we consulted only the female heads of household for this subject, whereas Benjamin had said you needed to take everybody in the group. So you need to detail the specificities as well. And so where are we? Voila. when I review methods sections, I have some questions that I pose in my head, and I think, okay, is the object that has been studied detailed adequately? Do we know what they were doing? Do we know how they did it? Are the methods described well enough to be replicated? So, you know, can I redo the method if I have access to that that population or those chemicals or the machine that did that? Can I do exactly the same thing? Do we understand what was done? Is the study design clear? Were there adequate controls? Were there treatments adequate? Were in the instance of perhaps looking at the impact of pesticides on something, were the concentrations of pesticides that were tested, are they realistic? Are they you know, related to what actually happens? And we come to the statistics. Are the statistical methods included? Are they appropriate? This is something that is, there is a phrase in English by an American author that was lies, damn lies, and statistics. So what he basically wanted to say was statistics, you can use them to say anything you want if you use them inappropriately. And this is why I really recommend discussing with somebody who is a statistician or who uses statistics well, correctly, to discuss with them how to treat your data, how to analyze your data. It's what I do, because you cannot be the expert of everything. And sometimes you think that a statistical test is appropriate and it's not at all. And sometimes you use the statistics and it will give you a response. Maybe you will be forced to reject your hypothesis, but maybe it's because the statistical test really wasn't appropriate. Or of course, maybe sometimes you accept your hypothesis and you used inappropriate statistics and the reviewer will say, no. You can't use that statistical test because you are violating the rules of it. You know, some of them you have to have certain, you have to respect certain situations for the use of statistical tests. Okay, so that is what I wanted to say about the methods section. Now, I think if you like, we can have some questions. Who is this conversation? Good. The difference between lasagna and spaghetti. Okay, you weren't listening when I described it in the first time, I think. So, the difference between lasagna and spaghetti. Spaghetti is when you know how spaghetti is, like Vietnamese noodle soup. They go in all different directions and sometimes you have to cut them up. Well, spaghetti is when you start talking about one thing. So, Barcholderia pseudomallei is a saprophyte bacteria that causes melioidosis. And then you would go and say something like 52% of our samples were positive. Okay, but what are you talking about? In Laos, this disease is endemic. Okay, bon, ça, the results. There was a statistically significant difference between river sediments and paddy field sediments. Okay, but it's going around in all different directions. You don't know where you are. This is what I mean by spaghetti. Lasagna is when you have Barcholderia pseudomallei is the pathogen causing melioidosis. This bacteria is endemic in Laos. Many rice farmers are affected by this work. We wanted to test if the bacteria was present in rice paddy soil. We found 52% of our samples were positive. We conclude that the bacteria lives in rice paddy soil. And so this is the kind of structuring that you're going for. That what I just said was probably an abstract, in fact. So you can see the structure. This is why I really like subheadings, because it makes it so much easier to structure. In fact, probably I would say the whole message about this training course is structure, structure, structure. That is probably the most important thing. So if you like, we can have some more questions. We can have a look at maybe some methods article, some method sections in different articles, if you want. We can, maybe if Benjamin would like to talk about how you treat methods in ethnological social sciences articles, how you guys see that, or anything else. It's up to you guys, please. So you can write your questions.
Yes, please ask any question you like. And I don't feel very comfortable to talk about how methods are presented in social sciences, in human sciences. Maybe Stefan have a more accurate opinion, because he has been working a lot on papers recently. Stefan, are you there? Do you copy, Stefan? Stefan? Yes, he's here.
Yes, I'm still here. I'm still hearing you. Noel, it's not so easy for me to answer this question, but what I will propose, focus on social sciences, is to for the trainees, is to have a look about all the publication available open source in the TAMDAO website. There's a lot of, we publish around 10 or 12 books, and some of them focus on how to build a survey on the field at the village level, for example, how you ask questions, how you keep the data, you organize the data. It's very complex thing. So it's not, and I was not prepared for this question. Excuse me for that. But just to escape, I will maybe ask the trainees to have a look on the tamdao.conf.com website, and they can choose different chapter of what we did the last 12 years on social sciences methodology. Is very rich, and I think it's quite easy to understand. This is my answer, Ben.
Okay.
Yes, thank you, Stefan. There is, you brought up a really important point, is about databases. And I think it's the way that we present and conserve data is extremely important, because we're all doing this work, and then when we publish it, it's like data that has been digested. But it's really important to maintain the databases with the raw data. And nowadays, we can do data papers, and it's one way of getting this data in its raw form known, published, and protected, because you have a DOE. Do you guys all know what DOE is? DOE is what in French you call a DOI, which makes me crazy, but it's DOE. It's a digital operator identifier.
Oh.
Thank you, Stefan, for putting it in the conversation, the TAMDAO conference. It means that your data is published and it is referenced to you and your co-authors, And when people use that data, they must cite you as the data source. It is a way of putting your data out there with a protection. You will talk about intellectual property rights on Thursday. And I really encourage you to look at these data papers and to see how they're used, because we all have data that we're sitting on. I am very guilty of this. And it's, you know, I'm starting to put it onto data websites so that other people can use it, because I know I will not have the time to publish the data. And because research, it's about sharing, in fact. And so I encourage this. This all goes into this new movement towards open science. Stefan said that the Tamdao conference, the data is open access. I really have a strong opinion about open access. I think we all need to do open access. I know it's not so easy for some of you guys because data has a value. For me in IRD in France, it's easy. My data has no value, no dollar value any. So it's like that. But I know it's some of you guys, it's how you finance your research is by selling the data, and I understand that. But we need to, I think we need to find a compromise on this.
Mm-hmm.
Yes?
I just want to add one thing. In one book publication, I do not remember the year, but maybe 2011 or 2012, we have a workshop focused on the complementarities between quantitative and qualitative approaches. And I think it will be very useful because our panel is very open, and it will be very nice to have a look on this publication just to see how it could be useful, but also how it could be difficult to have this kind of approach in social sciences.
Mm-hmm. Yes, thank you, Stefan. In fact, I work with health geographers. This is why I know about the semi-directive interview technique. And we are writing a paper at the moment with bacterial measurements in water, and we link it to health data. So that's, you know, data science, but we link it to the results from the semi-directive interviews. And it is one of the most difficult articles that I've had to participate in the writing of, because it's hard to bring those two together. You have the two approaches, but it's a great learning experience. And I think it's how we need to go forward more, is to bring those two together, because it would be much richer in terms of impact. So this is me on one of my favorite subjects, so I will not. Do you guys have any questions instead of me ranting on? No, I've tired you all out.
Okay, are you still there? Anybody?
Maybe
Hello, hello.
I'm here, but you are not thinking about me, right?
Yeah.
Yeah, okay, good. Are you asleep?
Ah, yes. I saw the semi-directive technique. It's when you ask questions. You don't, you ask questions that are quite directed. You know, what do you think about this water quality? Do you use this? Does it make you sick? This was the type of questions that were being used to determine why people were using a specific water source. It's in West Africa, where they have no access to drinking water or to sanitation. So this was the type of questions. That's why I was talking about them. I think also...
if there are no questions,
yes?
I can present you the next steps of our webinars, and particularly the homework for tomorrow. And actually, I have a bad news and a good news for you. I will start with the bad news. It's always easier to finish with the good one. The bad one is that you will have a little bit more work than usual today. But the good one is that tomorrow you won't have work. So it's you know, pretty good deal, I would say. For tomorrow, I will ask you to send a table or a schema, comment on dit schema? A scheme? No.
Conceptual diagram.
Yeah, a diagram or a figure or something graphic that you have used in the past in one of your paper maybe, or in a dissertation or whatever, and you will send it to the person which is after your name in the list, as usual. And this person will comment. So basically, you will send one table or one graphic diagram to the n+1. So it takes you 1 minute, okay? And the one you will receive, you just have to comment about it. You just have to say how, what is well done in this diagram, in this table. Is it, what is clear? What is less clear? So I'm in the learning process here, so I got it. So you must critique the table, not criticize, but critique it, which means in a positive way. Just give your opinion about it. What is well done? What is less well done? What is clear? What is less clear? Does it stand alone? Is that clear for everyone? Stand alone. It means, does it make sense by itself? Right, Emma? I'm not wrong?
Mm-hmm.
Okay.
With the legend, of course, but still, you know, the legend to the figure or the table and the figure, it means that you know what it's talking about without having to read the body of the text.
Mm-hmm. Mm-hmm. So don't worry, I will write this in an email later today, so you will have this information, but you can already, right now, we still have 8 minutes, so just dig into your personal archives and send to your n+1 in the list, send the table. And so that it is done, and then the one you will receive, you comment it and you send to your n-1 this time. The answer with me in copy, please. Always
Yes.
be in CC, otherwise I can't keep all this precious work you're doing. So this is for tomorrow. And
yes.
why you won't have work after tomorrow, I mean tomorrow, is because I'll ask you to do it for today, today for after tomorrow. And it is a little bit different, and you will have to join your national colleagues, because we need to have ideas about how the question of intellectual property and ethics and things like that are managed in your countries. So everybody from Vietnam will have to work collectively to give a short description of institutions or rules related to intellectual property and ethics in research. So it's a little bit more work demanding, I understand that, but it is very important, most of all, because I think it's not necessarily very clear how these things work. Also because it's important, and it's one of our objectives that you work together. And because of this distance learning methods, it's very hard. All the people who have participated to other trainings know that usually you work a lot in groups. but you know, with the situation, it's pretty hard to do it virtually. But at least here at national level, you can join forces. You design one of you to present the situation and the rules and regulations in your country. So I will write that also in the email. And this will be for Thursday. Okay. So if there are no more questions, I just have to thank Emma a lot, because I know it's hard to be at this time in the morning on your own in front of your screen. I know you manage that easily. It's much easier for you at 6 o'clock in the morning than at 11 at night. But still,
oh yeah,
you know, it's the kind of a lonely exercise when you just talk to your screen, and
it's strange. Yes,
it's really great. And if everybody is fine with that, I just have to thank everybody.
I have one point.
Yeah.
When you're looking at the figures and tables, think about what I said this morning about the figure in the PDF about the Becquerel area. You can use some of those ideas when you're thinking about the critique of the figure or the table, and what you would have maybe suggested should be different if you were the reviewer. Voila.
Okay.
This is it. So, yes.
This is it. So thank you, everybody. Thank you for being here.
Thank you.
And we'll talk tomorrow. And meanwhile, I will send you all your homework and great actions to do. Okay?
Have a good day. Have a nice day.
Have a good day. Thank you. Bye-bye, guys. Bye. I shall close the meeting.
C'est toi qui dois fermer, Emma.
Yes. Bye.